Right. Today we are going to learn about the genetic screening. Now, what is the meaning of genetic screening? If someone has a genetic defect, this defect is not visible from the outside because if we look a person gene, the chromosomes arrangement. So when they have this homologous chromosomes, example like this. If the dominant allele is a capital A and the recessive allele is a simple A, in this situation, dominant allele express. That means this allele express the character and the person is completely normal because the recessive allele is not expressing. And let's assume the recessive allele has a mutation. But luckily, this recessive allele is mutated and it can't express. Now let's see, this is from the father. And similar, we also get in the similar situation in mother. So unfortunately, mother is also having the same kind of genotype. That is a combination of alleles and capital A and the simple A. Now the simple A one again, mutated one, but it's not expressed, only the dominant one expressed, and then mother is also healthy. So both are fine and they do not have any problems. Right now, in this couple, if they want to have a children, they have no idea whether they have this kind of a defective gene. But if they are married and after that they're going to the doctor to see the conditions on then delivery, discussing the delivery and a regular checkup of the pregnant mother and all these things, the first meeting at the hospitals. The parents are also advised about the genetic problems because when they take the records of the mother, there is a question again, do you have any allergies? Do you have any genetic problems in your families? Do you have any genetic problems? And in this situation, maybe the mother and father both are aware there is a situation like this. But the problem is they are too late now. They can't do anything at the moment. So in this case, they have one option. This is called prenatal genetic screening. A prenatal genetic screening. Right, what is prenatal? Prenatal is before birth of the baby. So before birth of the baby, then they can check the genotype of the baby. So how they do that? There should be a special agreement with the parents and the hospital and the doctor, and they have to sign document, and they get all information from the genetic counselor and maybe after the doctor, and they have again a special timeline. They can't do at any time. So there are two types of genetic tests. One is amniocentesis. The other one is chorionic villus sampling. Amniocentesis and chorionic villus sampling. Usually the chorionic villus sampling is the first one. So this can perform between 8 and 12 weeks of pregnancy. And amniocentesis can be performed between roughly around after 12, up to 16 weeks. So chorionic villus sampling is the first window. If they miss the first window, they can go for the second window, amniocentesis. Okay, let's see what is the first genetic screening test, chorionic villus sampling. Right, in chorionic villus sampling, I abbreviated this one CVS, chorionic villus sampling. So how we can understand this chorionic villus sampling? It's like this. Let's say this is a developing fetus, and the fetus connected with the mother to the umbilical cord. So this umbilical cord make a connection with the mother side, and that is called the placenta. So this yellow line indicated the mother's tissue, and the white line indicated the fetus tissue. And the place where we can see this white color area is called chorionic villus. So now the idea is to take sample from the chorionic villus. So we take a small piece of tissues which contain cells of the fetus from the chorionic villus. It's not that a simple procedure, because it's a little bit of risk for the fetus. Always this kind of a test has a small percentage spontaneous abortion. Right. How we can understand this test? It's like this. So we go step-by-step guideline performing this test in this video. Okay, the doctor carefully observe the patient's health conditions, and everything is ready. Then the doctor can decide the exact time for this chorionic villus sampling. And first thing is they have to do ultrasound. So what is ultrasound? They can check. They can check this fetus. You can see here the face of the fetus, and this is the developing fetus. So they have to carefully observe the fetus and its position and everything. And then they have to locate the placenta. So first thing is, I will go for the preferred method. The first thing is, you can see the probe. So this probe scan the fetus. So the probe placed on the stomach, the belly of the mother, and then through the skin, it can get the images. So then these images are on the screen, so the doctor can see the exact position of the baby. And then doctor insert a special a catheter through the cervix, and then they can aspirate. That means take some sample by a suction method from the placenta. So the procedure is 1 to 2 hour, one to 2 hour procedures. And then after that, the mother can stay a little bit longer in the hospitals to see any kind of a complication of the procedure, to face that kind of a complication. And then the test report will come after a few days. They can check the result. So that's a chorionic villus sampling procedure. Right, let's see what is the basic steps. One is ultrasound, scanning, then through the catheter, Small sample is taken from the core inequality. CV is the core inequality, right? Number 3, then test the sample. So they can analyze DNA to find any known recessive alleles. And if they find the combination of the homozygous recessive conditions, then this confirm there is a chance of having a genetic defect in the baby, right?